The last biological molecule that we're going to look at is lipids. Unlike carbohydrates, which are made up of monosaccharides, and proteins, which are made up of amino acids, and nucleic acids, which are made up of nucleotides, there's not a single repeating unit for lipids. They are made up of common subunits, like glycerol and fatty acids, but there's no true monomer for lipids. They're actually a very diverse group of molecules that are mainly hydrophobic, and they contain hydrogen and carbon primarily. They're completely insoluble in water. That's what is meant by being hydrophobic. Water and lipids, like fats, don't mix. They are formed by linking monomers into polymers, so we do still use dehydration synthesis to put them together. And lipids, kind of like proteins, have a variety of functions. Energy storage is key, so they're kind of similar to carbohydrates. But lipids are for long term energy storage, whereas carbohydrates are for readily available energy use. We also have things like waxes that provide waterproof coating on plants. We have phospholipids that are primary component of cellular membranes, and hormones, which are derivatives of cholesterol and examples of steroids. So there's a lot to look at when we talk about lipids. They fall into three major groups: oils, fats, and waxes, phospholipids, and steroids. So we're going to be looking at these three groups. Phospholipids are major components of cell membranes. Steroids include cholesterol, and many steroid hormones are just derivatives of cholesterol and their shape. So let's start with the first group: oils, fats, and waxes. So they contain only carbon, hydrogen, and oxygen, and they contain at least one fatty acid chain. One of the most common types of fats and oils that we're going to look at are triglycerides. So they contain three fatty acid chains. Here we can see dehydration synthesis putting together a glycerol molecule, which is a sugar alcohol, and three fatty acid chains. The fatty acid chains do have a carboxyl group at the end, and that's where we're going to get the OH, and then we're going to get an H from glycerol in order to remove water 3 times to put these molecules together. That's going to give us the structure called triglycerides, sometimes neutral fats. Fats and oils are used primarily as energy storage molecules. They contain twice as many calories per unit as carbohydrates or proteins. Fats we find mainly produced by animals, and oils mainly produced by plants. So let's talk about the different types of fats. We have saturated fats and unsaturated fats. Saturated fats tend to be solid at room temperature, whereas unsaturated fats tend to be liquid at room temperature. Saturated fats are solid and they have single bonds. That's a lot of S's put together. Saturated fats that are solid at room temperature include things like butter. The reason that they have a solid formation at room temperature is because the single bonds allow all these fatty acids to group together, and when chemical bonds and molecules are close together, they are more likely to form solids. So here is a fat that we consider a saturated fat. Again, like butter. Saturated fats are solid at room temperature due to the single bonds between carbons in the fatty acids. So let's review saturated fats. Saturated fats are solid at room temperature, don't have double bonds, they only have single bonds. And so that's kind of a quick overview of what you should know about saturated fats. Unsaturated fats, on the other hand, are oils, or generally called oils. They tend to come from plants. They have fewer than the maximum number of hydrogen attachments to carbon because they have double bonds between their carbons. And that's going to give their fatty acid tails a bend or a kink. And that's going to mean that they can't fit closely together and are more likely to be a liquid than a solid. So examples of unsaturated fats are things like avocado oil. They're liquid at room temperature, and they do have double bonds in their fatty acid tails. What about waxes, though? We looked at fats, we looked at oils. What about waxes? They are chemically similar to fats, but a lot of animals like enzymes to break them down. They're mostly highly saturated and solid, especially in outdoor temperatures, but the big thing that is important about them is they are water repellent, or generally they are. So they're found in the leaves and stems, they protect plants, they prevent waterlogging, they're found on bird feathers and in honeycomb, so they're really helpful as being a water repellent. And then we can't forget phospholipids. So we're moving on to that second group of lipids. Phospholipids make up plasma membranes, also known as cell membranes. Usually a plasma membrane contains 2 layers of phospholipids, so it's called a phospholipid bilayer. They do somewhat resemble oils. There's usually at least one fatty acid tail that has double bonds and is unsaturated. Here's the basic structure of a phospholipid molecule. It has 2 dissimilar ends. Another way to talk about a phospholipid is to say that it's amphipathic. One end behaves a certain way, and the other end behaves completely differently. Its polar head, where we have a variable functional group attached to a phosphate group, it is going to be hydrophilic, and it likes to hang out around water and other polar molecules. The other side, which has fatty acid tails, is hydrophobic, and it does not like to hang around water. So that creates 2 layers in order to keep these areas where they want, and that creates a cell membrane. It actually provides a lot of structure for how a cell membrane behaves. At the end of this PowerPoint, we'll look a little bit more at that structure. But let's first talk about steroids. Steroids contain at least 4 fused carbon rings, which is the base structure of cholesterol. If you modify that and add functional groups, you can get other steroids like steroid hormones, estrogen, and testosterone. Cholesterol itself is a vital component of membranes and are in animal cells in different ways. Steroids are always going to have that 4-ring structure. For example, here we see cholesterol, and if you modify that long functional group, you get estrogen and testosterone. But what's similar among all of them is they have that 4-ring structure. But before we end looking at biological molecules, we want to make sure that we go back to phospholipids, because further chapters are going to look at cells, and a cell membrane is a major component of cells. It gives cells an inside and an outside. And the way that we do that is we use this amphipathic phospholipid molecule to create a bilayer. This bilayer has the polar head with the phosphate group in phospholipids arranged facing the inside of a cell and the outside of a cell. That's where we have the watery environments. So the hydrophilic polar head of phospholipids tends to face those environments, whereas the nonpolar hydrophobic tails are sequestered away from these watery areas, and they're protected, but also give that cell membrane a certain chemical and physical property, where it's hard for things that are polar and charged to go straight through the membrane. That actually looks something like this when we look at the orientation of water and the phospholipids within the cell membrane. The symbol for a phospholipid, in order to shorthand it, is generally a circular head that is hydrophilic, and then 2 lines to represent the 2 hydrophobic tails. Again, here we're viewing a cell membrane orientation with the phospholipids having their tails in the middle and their heads on the outside, and that's representing the outside of a cell and inside of a cell. More on that when we talk about cells later.